Good morning, dear friends. Myself, Vivek Jedhe, Head and Assistant Professor, Arts and Science College, Pulgaon. Today, we learn methods of transformation or transfection. Transformation means uptake of DNA from surrounding. DNA can be taken as DNA fragment or DNA plasmid. The bacterial cell will take DNA fragment, will either integrate with bacterial chromosome or degraded. When it is integrated into bacterial chromosome, it is known as stable transformation, and when it is degraded, then it is known as unsuccessful transformation. Many times naturally bacteria take DNA plasmid from surrounding. This uptake of plasmid is called as stable transformation, as you are seeing in figure. There is a difference between terms, that is transfection and transformation. Transfection is the process of deliberately introducing naked or purified genetic material into eukaryotic cells, while transformation is direct uptake and incorporation of exogenous genetic material from its surrounding through the cell membrane. So, transformation is broader concept than that of the transfection. Transfection is done purposefully, so transfection is an artificial process where the introduction of foreign DNA into mammalian cells takes place. Both transformation and transfection uses various kinds of method of transformation, that is calcium phosphate precipitation method, microinjection technique, electroporation, etc. There is another term called transduction. Transduction is a method of gene transfer in which the genetic material is deliberately introduced into another cell with the help of viral vector. When viruses are used for transformation, then it is known as transduction. This is a biological method, while electroporation and microinjection techniques are non-biological methods. What is the history? Transformation was first demonstrated in 1928 by British bacteriologist Frederick Griffith in Diplococcus pneumonia. In 1994, the transforming principle was confirmed that it is DNA by Avery, MacLeod and McCarthy. In 1980, the transformation using electroporation technique was developed, and in the same year, the transformation can be done with the help of gene gun technique by John Sanford. There are common methods of transformation or transfection, that is chemical based method, physical based method and biological based method. Chemical based methods include calcium phosphate precipitation, liposome technique, magnet mediated transfection, while the physical methods have electroporation technique, microinjection technique, etc. The biological based methods include only one technique, that is viral based transfection. Let's see what is calcium phosphate mediated DNA transfer. In this process of transfection, administration of isolated DNA, that is near about 10 to 100 microgram, with the solution of calcium chloride and potassium phosphate, and thus calcium phosphate is formed. This calcium phosphate is incubated with DNA, and that will form a precipitate, and that precipitate is taken up by bacteria with the help of endocytosis. So, the bacterial cell uptake the DNA with the help of endocytosis, and the DNA become the part of bacterial chromosome. This method is used for the transformation of prokaryotic cells mainly. Let's see the flow chart. Take the growing E. coli cells, incubate it with the calcium phosphate or calcium chloride, and for up to 12 to 24 hours, and recombinant DNA will added into the plate. Then the cells plated in a suitable medium for growing purpose. Then screening for the transform cells, and the transform cells are diluted and recultured in suitable medium. This method is also known as calcium chloride method of DNA transfer. Next, that is physical based methods. Physical based methods are mainly used nowadays because they are having more successful rate. One of them is electroporation technique. Electroporation was first reported for gene transfer in mouse cells. The technique is often used for cell types such as plant protoplast. Electroporation uses electrical pulse that it creates a temporary pores in the cell membrane with the help of simple electrical pulse. Let's see how. Suppose there is a cell membrane, and that cell membrane is exposed to simple electrical field, very low voltage. Then that will create a pores, and that pores are responsible for uptake of DNA from surrounding. This is the simple principle of the electroporation technique. Here you will observe in diagram, that is how the electroporation technique is done. Electroporation, that is general idea behind electroporation, is by applying short electrical pulse to the cell membrane, conductivity and permeability is altered, and that is responsible for uptake of the foreign DNA material. The hydrophilic pores are mainly formed in the membrane. You all know that the membrane has both hydrophobic and hydrophilic properties, but here, due to electroporation, only hydrophilic pores are developed, and that do not create any problem with the cell turgor pressure. In this technique, we will take only the cells in log phase. This is the procedure of electroporate, how electroporate is formed, and how it will useful to for the transformation. This is a electroporator. It just simple look like your PCR. Electroporation, that electroporator which is used, that is having, you are seeing in the diagram, that it having a power supply, then it having a cuvette, then having a electrode, and having the cells which are used for the transformation purpose. What are the advantages and disadvantages? Both cells and tissues can be transformed with the help of this technique. Mind it, when we are using eukaryotic organisms, we require tissue to be transformed. So this technique is useful because both in prokaryotic as well as in eukaryotic. The efficiency of transformation depends upon the plant material or the material which is used. Next, disadvantages only that only 40 to 50% incubator cells receives DNA, and less than 50% transform cells get survive. So that is the biggest disadvantage of this technique. Let's see what are more advantages. That it is fast, rapid technique, biological structure and functions become intact, easy to perform, high efficiency rate. Another disadvantages is that cell mortality and low efficiency. You all know that cloning. You all know that the cloning technique is developed, and that is responsible for development of the shift that is named as dolly. That dolly is developed with the help of cloning technique, and in cloning technique the microinjection technique is used. This microinjection technique is also an advanced technique of DNA transfer. In this microinjection technique, the DNA solution is directly inserted inside the nucleus of the cell. So it has high efficiency. Advantages that frequency are stable, the methods are effective in transforming the primary cells. Disadvantage that it is very costly and requires skilled workers. So this method requires skilled workers that will be helpful. Here is a diagram in which you can easily understand what is the microinjection technique, what is electroporation technique, what is viral transduction, what is the delivery of DNA with the help of lipid mediated transfer, what is the polymer transfer, etc. And you will clearly understand the concept of all these transfer techniques. Here is a video. You can see video link. You can take the screenshot of that and watch on YouTube. Here are references, that is molecular biology by Upadhyay and Nath, molecular biology by David Freifilder, cell biology, genetics and molecular biology. I mainly prefer you, that is the molecular biology technique, which is my molecular biology book that is written by David Freifilder. That is most authentic book. Some suggested visits with YouTube are there. You take screenshot of this. Thank you.